Welcome to The Explainer. Today, we're diving straight into the invisible ecological domino effect triggered by chemical pollutants. If you're an undergrad studying environmental science, conservation, or biology, you absolutely need to know this stuff. This is a foundational look at how microscopic inputs literally rewire global ecosystems from the bottom up. So let's look at exactly how these unseen chemicals move through our world.
Here's our quick roadmap for today. We'll be breaking down three major pollutant classes and their cascading impacts on biodiversity, starting with heavy metals, moving into agricultural pesticides, and wrapping up with organic and sensory pollutants.
All right, part one, the pollution threat and heavy metals. Let's get into it.
So we're talking about naturally occurring elements that get dangerously concentrated because of things like industrial activities and mining. Common culprits like arsenic, aluminum, lead, cadmium, and mercury pose a really unique threat. And the reason they're so dangerous is because of direct toxicity. They literally bind to crucial enzymes and totally disrupt foundational cellular functions. Even at barely their trace concentrations in aquatic environments, exposed invertebrates experience severe developmental abnormalities and their reproductive success just tanks. We aren't just talking about making an animal a little sick. These metals fundamentally disrupt an organism's ability to grow and survive at a molecular level.
Now, as environmental science students, you absolutely have to know the difference between how these metals are stored and how they travel. The distinct difference here is between bioaccumulation and biomagnification. Bioaccumulation is all about the individual. It's when a single organism absorbs the toxin over its lifetime and it builds up in that specific animal's tissues. Biomagnification, on the other hand, is the food chain effect. It's what happens when that toxin actually multiplies in potency and concentration as larger animals eat smaller ones.
To really see biomagnification in action, look at this progression. See how the red toxin markers multiply? We start way down at the bottom with phytoplankton absorbing ambient pollutants. Then zooplankton eat them, concentrating the dose. Next, herring eat the zooplankton, salmon eat the herring, and the toxins just compound exponentially. By the time you reach an apex predator like an orca, the toxicity levels have skyrocketed, creating immediate severe risks to their reproductive viability and overall health.
And the scary part is, chronic exposure to these metals creates massive ripple effects beyond just predator health. It completely alters community structure. You'll see tolerant species survive, while sensitive ones just vanish, completely flipping the ecosystem's dominance. It also physically degrades habitats. Contaminated wetland sediments, for instance, directly harm benthic organisms, the creatures living right at the bottom, which then ends up starving the water birds and fish that rely on them. Plus, and this is huge for your genetic studies, heavy metals can cause severe DNA damage and alter gene expression. That means long-term genetic consequences that strip a species of its ability to adapt to future changes.
Okay, let's keep it moving to part two, pesticides and food webs. We engineer insecticides, herbicides, and fungicides to do one job: protect our crops. But here's the problem. While pesticides are supposed to target specific pests, their direct toxicity ends up hitting a massive list of unintended non-target organisms. We're inadvertently wiping out crucial pollinators like bees and butterflies, natural predators like ladybugs, and the fallout doesn't stop there. It extends to insectivorous birds, amphibians, and mammals. We spray a chemical to kill a specific weed, but the poison doesn't discriminate.
For a really highly relevant real-world example, we have to talk about neonicotinoid insecticides. Now, you might think a bug needs a lethal dose to be affected, right? But actually, sublethal exposure is just as devastating. These chemicals specifically bind to acetylcholine receptors in the insect's central nervous system. They overstimulate and eventually block them. This neurological disruption completely messes with the navigation and learning abilities of crucial pollinators like honeybees. If they can't forage efficiently, the entire colony suffers.
And when herbicides wipe out plant diversity and airborne pesticides wipe out flying insects, you disrupt the entire intricate food web. It really is like an ecological game of Jenga. You pull out the foundation, the plants and the insects, and suddenly the insectivorous birds and bats have absolutely nothing to eat. Their populations collapse. Ultimately, this disruption starves dependent species and drastically shrinks overall biodiversity.
And here is the ultimate tragic irony. We often make the original problem so much worse by triggering an insidious evolutionary arms race. Think about it. Step 1, we apply a pesticide. Step 2, a few naturally tough, genetically resistant pests manage to survive it. Step 3, those specific survivors reproduce, passing down those hardy genes. And Step 4, that resistance spreads like wildfire, creating a superpest population that demands even harsher, broader spectrum chemicals. It just restarts the whole destructive cycle all over again.
Which brings us to our third and final section, organic pollutants and beyond.
This is a massive umbrella covering everything from complex pharmaceuticals to everyday plastics. We're looking at persistent organic pollutants, or POPs, which are pouring into our ecosystems from a staggering array of sources. Solid waste landfills, industrial processing, agricultural runoff, and even the daily vehicle exhaust fumes from your commute. The key word here is persistent. These carbon-based chemicals do not break down easily. They literally linger in the environment for decades.
And the impacts of these persistent chemicals are profound. They are notorious for hijacking wildlife endocrine systems, which means they scramble an animal's hormone production. This leads to those shocking, well-documented historical events you've probably read about, like DDT causing severe eggshell thinning in birds of prey, almost wiping out the bald eagle. We're also seeing the feminization of male fish because of synthetic estrogens from pharmaceuticals getting into our waterways. And on a larger macro scale, agricultural runoff packed with organic compounds overloads aquatic ecosystems with nutrients. This triggers intense algal blooms and massive oxygen depletion, a destructive process known as eutrophication.
But as we study all of this, it is super vital to recognize that pollution isn't solely chemical. We have to look at the whole shebang and contrast those toxic inputs with physical and sensory pollutants. We face physical pollution like macroplastics causing marine entanglement and starvation. And then there's sensory pollution, artificial light at night totally disorienting nocturnal animals and messing with their migration, and noise pollution, drowning out crucial mating calls. The footprint we leave on biodiversity is multisensory and structural, not just molecular.
So, as we wrap up this explainer on the cascading impacts of our chemical world, I really want to leave you with a provocative thought. As undergrads, you are the ones shaping the future of conservation. Look at the evidence we just walked through. From the single-use plastics you throw away, to the artificial light coming from your dorm, to the unseen heavy metals inside your smartphone, how might your daily footprint be the very first domino in a distant ecosystem's collapse? Thanks for hanging out with me today, and keep questioning the invisible connections that hold our world together.